This is a lymph node. So you're looking at a section through a lymph node. And there are several things you should be able to recognize. One is this blue stained structure right here, which surrounds the organ. What is it? That is the capsule. Okay? Made of denser fibrous connective tissue that supports and protects the organ. It is not unique to lymph node. It's found around many organs. So that's the capsule. Okay? There's a space just below the capsule. You can see right here. What is this called? Subcapsular sinus. And again, remember, a sinus is a cavity or space, like the sinuses in your skull. Remember, these are cavities in the bone. Okay? You'll also notice there's an extension from the capsule that projects in the interior of the organ. These would be trabeculae. Trabecular singular, trabeculae plural. The spaces around the trabeculae are trabecular sinuses. Okay, so lymph is brought in by what vessels? What vessels feed lymph into the lymph nodes? The afferent lymphatic vessels. Okay, penetrates the capsule. The lymph then flows along here in the subcapsular or cortical sinus and then follows the trabeculae in. In other words, along through the trabecular sinuses. Then goes into the interior region here of the organ. What do we know this as? This is the medulla. And these spaces or cavities in here, what are they called? Medullary sinuses. These purple areas of tissue around them are called what? Medullary cords. Okay? And really they're just collections of cells. What kind of cells? Okay, lymphocytes, because remember it's a lymphatic organ, always going to have lymphocytes there. They become sensitized by foreign antigens passing through what they're exposed to. There's also another type of cell here, because remember, what does a lymph node do? Not only serves to sensitize the lymphocytes and stimulate the immune system, but what else? It's filtering the lymph. And how does it do that? By means of phagocytic cells called what? Macrophages. Where do macrophages come from? From monocytes that have left the blood. Okay? So you got the big eaters, the macrophages, to gobble up the debris. Okay? And you got your lymphocytes here to become sensitized when they come in contact with foreign antigens. And again, what's the fluid passing through here in these sinuses? Lymph, not blood. Please be careful. Think about what the organ does. It filters lymph, the lymph nodes, not the blood. Okay? Out here in the cortex, there's some very distinctive features. What's this? That would be a secondary follicle. You can tell because it's got the light region called germinal center, surrounded by a darker stained region, corona. And these are all lymphocytes here. Okay, these are developing lymphocytes here. These are the older lymphocytes. But eventually they will come down into the medulla and they make up these medullary cords. They'd be found in here. Okay? And then the lymph is drained from the lymph node by what vessels? The efferent. So it's A before E. A brings the lymph fluid in, lymphatic vessels, and efferent vessels drain it. Okay? We got a lot of secondary follicles, but here and there, you're going to see some here that are kind of uniform color throughout, and these would be the primary follicles. They're few and far between, and they have no germinal center. Okay? Have we left anything out? Okay, I don't think so. Any questions you have on this?
We just passed through a follicle, right? With the top,
Well,
top all the
we
way
passed through a
around.
bunch of them. There are a couple secondaries here. There's another secondary right there.
Okay.
Yeah, and
And the one
remember
on
the
the left?
follicles are in what region? Only in the cortex. Okay? And if you look at it, the cortex is much denser. You can see it here very nicely. Is much denser than the interior, the medulla. So this is all cortex out here. You can see all these follicles. And here's the medulla. It's much more diffuse because you got this is where the sinuses are. The medullary sinuses, and then the purple tissue here, that'd be the medullary cords. And again, what's being filtered here? The lymph. Okay? By the way, these blue things here, that's part of the trabeculae, and also some of these things here are blood vessels, which I don't expect to recognize yet, but next unit you're going to be picking out blood vessels and tissue. Any other questions? All right, let's look at another lymphatic organ. And this is the spleen. Now the spleen again has a capsule about it, but you may or may not see on some of the slides you looked at the capsule. Because remember, the spleen's a fairly large organ. You have identified on those models. This one has a capsule along only one side. And that's what this is. Again, it's a dense or fibrous connective tissue membrane that surrounds and protects the organ. And extending from that, just like we saw in the lymph node, what are these? Trabeculae again. And basically an extension of the capsule that projects into the organ and provides further support. Now the spleen has two regions. We see one right here. What is this? This is the red pulp. It is not the cortex. There is no cortex or medulla in the spleen, so be careful on that. You either got red pulp or you got white pulp. And this is red pulp. Red pulp is characterized by having all these little cavities in it. And what are these cavities called? Okay, probably the easiest thing to remember is splenic sinus. Okay, do not call them medullary sinuses. There's no medulla here. Otherwise, it's going to be a red line through your answer, okay? Medullary sinuses are in the lymph nodes, not in the spleen. So you may call these splenic sinuses. You may also call them venous sinuses. Or if you want to be fancy, you can say splenic sinusoid, because it's actually a type of capillary, very porous capillary, that red blood cells can actually cross the walls, which is unusual. They're also called venous sinusoid. So 4 names you may use and get credit. 1 you may not. That's medullary sinus. Okay, so venous sinus, venous sinusoid, splenic sinus, splenic sinusoid, all acceptable. Okay. And out here, these cords, tissue around it, what do they call it? Splenic cords. Or if you want to be fancy, you call them Billroth's cords. And what kind of cells do we have here? What kind of cells? What? Erythrocytes? Not in the cords.
Oh.
Lymphocytes. It's a lymphatic organ. Should be a given, okay? We got to have lymphocytes, all right? So we got lymphocytes here. What else? Macrophages. What's the spleen doing? It's gobbling up worn-out red blood cells and debris in the fluid passing through in these sinuses. And what is the fluid? It's blood, not lymph. So be aware of what the organ does. The lymph nodes filter the lymph. The spleen filters blood. Okay? Takes debris out. It takes out worn-out red blood cells and also a lot of microorganisms that may be in the blood as well. Okay? And here again is one of the trabeculae, which you can see. So that's red pulp. The other region of the spleen, which is should be fairly obvious, is called the white pulp. And it sort of looks to me like they're little islets surrounded by a sea of red pulp. So this is the white pulp. And what do you have here? You basically have a secondary follicle, right? Like you saw in the lymph node. But it's got something else associated with it. This little thing right here. That's the central artery or central artery. It's a small artery, small blood vessel, bringing in blood. You can see it here. So again, if you get the pointer on this, as you identified, you should say central artery or central arterial. The region here of which it is a part is the white pulp. Question?
Yes, on the exam tomorrow,
You what?
on the exam tomorrow, like, are you gonna, because it'll be a little confusing with the red pulp and the
I think, what's the reading of the other way? Okay. I point this out and say, what's the structure? Indicate it, and indicate the structure. I say, okay, it's part of what region? And you say red pulp. If I have the pointer on this thing, don't say white pulp. Say central artery or central arterial, which is a small artery. Okay, and I could say, okay, it's part of what larger structure region? That's the white pulp. Okay? And it's fairly distinctive. Like I say, I'll give you good examples. There shouldn't be any guessing if you've seen these things. Some of those slides are better than others. And I'll take out the good slides. Like this. Okay? So if you've seen them, you shouldn't have any trouble. Here again, white pulp. This is also white pulp down here. This one's not been cut right through the center. That's why you don't see the whole thing. You only see part of the germinal center. Remember, these are 3-dimensional structures. You just got a thin 2-dimensional slice. Here's another region. There's your central artery again. There's your follicle. The whole region again, white pulp. Okay. Any questions?
Say that one more time, I couldn't hear you.
Say it one more time.
Yes, I can't hear you.
I said, there's your central artery, little pink donut. Okay? The region of which it is a part is the white pulp. And there's your secondary follicle. Okay? What else does the spleen do besides get rid of worn out red blood cells and filter debris from the blood? What else? I could ask for several functions.
Stores platelets.
What else?
Stores platelets and stores iron.
Okay, it stores iron, because remember it's breaking down those red blood cells, breaking down the hemoglobin. And it stores iron to be recycled.
Immune
And
surveillance.
that's why some of you, when you look at those slides, you had that brownish stuff in there, in the tissue. That was iron, stored in the macrophage that'll gobble up the hemoglobin. What else? What's it do as far as the fetus is concerned?
It's where red blood cells.
It's the site of red blood cell production, but only in the fetus. Later, that's taken over by the red bone marrow. Anything else? How about platelets? Anything to do with platelets? It's a platelet reservoir. Okay? And of course, you have lymphocytes proliferating here too, and they become sensitized. Okay? So know these functions. I can't stress that enough. There'll be a lot of questions on function. All right? Don't freak out. Well, you didn't say that. I'm saying it right now. Okay? Not that many models, not that many slides, so a lot on function on this test. Next test will be a lot of models, okay, and fewer slides. For those who don't like slides, you'll appreciate that. But there'll also be pigs and hearts on that test, so makes up for it. Any other questions on this? All right, let's look at another lymphatic organ. That's the thymus. What's the function of the thymus?
T lymphocyte maturation.
That's the site of T lymphocyte maturation, and only T lymphocytes. Okay, it's exclusive. You're only admitted if you're a T lymphocyte. B lymphocytes excluded. It's like a privileged country club. Okay, you've got a membership. All right. So, site of T lymphocyte maturation is what I want. Don't say college or finishing school. Right? That's what maybe amounts to. All right. Now you've got something again surrounding the organ. We've seen before. Kind of torn away, but what is this? That's the capsule again, connective tissue that supports and protects the organ. The thymus itself is kind of divided into little sections, you can see here. What is this? It's a lobule, okay? Small lobe. And you can see, very obvious, it's got a darker stained outer region. What do we call that? That's the cortex. The lighter inner region that's lighter stained is the medulla again. And then something very distinctive, found only in the thymus, and it's in the medulla of the thymus. You can see them scattered around here. What are these? You call them thymic corpuscles or Hassel's corpuscles. What is their function? And don't say function unknown. That's what your photo atlas says. But they now know there is a function. What do they do? Okay, they aid in the maturation process. So the site of maturation is the thymus, but it's these thymic corpuscles. They release chemicals that are important in the maturation process of T lymphocytes. So your T lymphocytes, like all blood cells, start out life in the red bone marrow. Then they very quickly migrate out to the thymus. There, they don't contact any foreign antigens, okay? They're maturing. They're getting ready to, getting prepared to, okay? Then when they leave, they're called naive, and now they're ready. So they go out to these various lymphatic organs, then they contact foreign antigens, then they become sensitized and do their thing by producing new cells. Okay? These cells start development again in the red bone marrow, then they go directly out to lymphatic organs like the lymph nodes, spleen, and so on, and they directly contact foreign antigens and do their thing, which is what? Antibody production. Okay? Any questions on any of this? Yes?
What is the Hassel's corpuscle again?
What are they? Want me to point them out?
Yes.
Okay. This is pink things. All over. Okay? And again, they're in the medulla, the light stained region, the interior of these lobules. And very distinctive for the thymus. So by the slide cover, you say, hey, I know what that is. That's a thymus. Okay? But you'll have them later. You don't have to freak out about that. But you better know what these things are. Now, if I have something that's on the label, well, obviously I'm going to cover the label. Okay? So don't say, well, you said all are going to be uncovered. Not necessarily, but most will. In fact, sometimes the question will say, in the thymus, blah, 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 or in the spleen, or in the lymph node. Okay, so there shouldn't be any question as to the organ you're looking at, but you need to know structures within the organ. All right, one last thing. What you saw again yesterday. Now, remember there are lymphatic nodules or follicles in various parts of the body. And that's part of what we call what? The MALT, which stands for mucosa-associated lymphatic tissue. Not mucus, but mucosa. And the mucosa is a type of membrane. It's a mucous membrane that lines many of the cavities in our organ systems. The GI tract, our respiratory tract, urinary, and reproductive. All lined, tubes in there, all lined with mucosa. You're going to see a lot of it, okay? Mucosa always has an epithelium and then some connective tissue beneath it called lamina propria, which we'll talk about later. Okay? But here in the small intestine wall, we have some examples of MALT, and they have a specific name, which you should know. It's on your handout. Okay? And these, again, are collections of lymphocytes. You can see it very distinctly. Kind of purple and granular in appearance. What are these? Peyer's patches, okay? The Peyer's patches. And they're in the mucosa here of the small intestine wall. And they actually extend down to what's called the submucosa. What are they? Collections of lymphocytes. That's what it is. And remember what lymphocytes look like? When you look at those blood smears, what are they? Big purple nucleus and very little cytoplasm. When you got them in tissues, stained, they often look like little purple granular areas. Okay? In this case, Peyer's patches, part of the MALT. Okay? Any other questions? Okay? Shouldn't be that bad. Should all get an A. Right? And again, make sure you look at the models. They can identify the tonsils. Can I borrow it for a moment here? Everybody see the tonsils? So there are no surprises here. Okay, here's a half-inch model, lymphadenal and coronal. Right on here, this is part of the ball. You got a tonsil up here in the upper part of the pharynx or throat called the nasopharynx, and this is called the pharyngeal tonsil, sometimes referred to as the adenoid. Okay, you can't see that when you open it